right, so let me give you an intro. Thank you so much for joining us today. So Professor Ellen Roche is a Latham Family Associate Professor at the Institute for Medical and Science and the Department of Mechanical Engineering at MIT. She directs the Therapeutic Technology Design and Development Lab. She completed her PhD at Harvard School of Engineering and Applied Sciences. I think I just missed you. I'm sorry. And then she did a postdoc at the University of Galway. So her focus is on applying, tech to cardiac devices. She spent 5 years in industry doing this. She has an amazing, array of tech that is directly applicable to truly benefit lives, which is amazing for roboticists to be able to say. She has a number of awards, including the Fulbright International Science and Technology Award, the Wellcome Trust Seat Award in Science, the National Science Foundation Career Award, and a number of more, including the NIH Trailblazer Award, which is very impressive. So we're super excited to have you give a talk today. Please take it away.
Yeah, thank you so much for that kind introduction, Professor Peterson. Yeah, it's a pleasure to be here today. And first of all, I'd like to thank everyone for their flexibility with a little bit of an earlier start to my talk than originally planned, but very much appreciated. So I will get started, and I'm just going to focus on two topics today. And I should be wrapping up by, I guess, noon here and 4 p.m. there with the change in time zones. Okay, I will jump right into the talk. So yeah, I'm going to talk about developing embodied intelligence into active organ assist devices and enhanced benchtop simulators. So I'll start by kind of describing what drives a lot of the research in our lab. We're really interested in moving organs, these cyclically dynamic organs that really sustain life. They are fascinating to me because they're, we're sitting here, I'm pretty still at the moment, but my diaphragm is shown here and my heart is moving constantly. And these two examples are muscular pumps. The diaphragm drives airflow in and out of the lungs, and the heart pumps blood around the body. And these, drive at a different scale, kind of drive alveoli to fill with air or valves to open and close. So it's just really fascinating how this all works without us even thinking about it. So our mission is both to enhance these cyclical organ functions and augment them when they fail, but also to recapitulate how they work on the benchtop in the lab. And I'll kind of give an example of a research project in each of these kind of research strands today in the talk. This is kind of a typical medical device design linear process here, where we work with industrial and clinical partners to design something. We simulate and test it and then translate it. And ideally, we have industry adoption, we commercialize our devices and clinical adoption. However, we spend a lot of time in this loop here where we're simulating and testing. And it's really here that we do a lot of really research focused topics, and we can really then establish insights into things like the biomechanics of pathophysiology and really understand better the mechanical influencers of disease and feed that back into designing better devices, feed it back into the clinic, and feed it back to our industrial partners, maybe with better test beds to test devices. So my overall research is kind of divided into 3 different sub topics or research strands: mechanical augmentation of organ or system function, structural or biological repair of tissue at a more local tissue level, and then multimodal simulations of the biomechanics, all of these cyclically dynamic organs. Today, I'm just going to focus on a case study, or 2 case studies from topic 1 and 3. And as I go through these kind of case studies, I'll talk about why we do it, exactly what we're doing, the team that's doing it, how we approach the problem, and what is the impact and the future work in each of these case studies. Okay, so the first topic is mechanical augmentation of organ and system function. And here I'm going to talk about work that was done as part of the Trailblazer award that Professor Peterson mentioned. This is an implantable ventilator that assists the diaphragm and also mimics the biomechanics of breathing. The clinical problem here is that the diaphragm can fail. The diaphragm, as I mentioned, is a major muscle that drives pump function for breathing. But in conditions where there's muscle weakness or degenerative muscular disease, the diaphragm can also become weakened. It can also become paralyzed. So in these cases, it's just not able to move and generate that negative pressure in the thoracic cavity to draw in air. You know, when this is at a late stage, at end stage respiratory failure, some of the treatments, as you will know about from the recent pandemic, there was a lot of press around them, are artificial mechanical ventilation. And this is typically positive pressure ventilation where air is pushed into the lungs. It can be non-invasive or invasive. You can have a mask or a tracheostomy, but in both cases, it significantly affects quality of life, and things like speaking and swallowing and eating become more difficult. So we saw this unmet need for an implantable ventilator that could mimic the breathing mechanics, but also preserve key aspects of quality of life. This is kind of a schematic of how we envisioned this implantable ventilator to look like. So we'd have actuators over the diaphragm, which would augment the function of that diaphragm or muscular pump. They would be connected to an external control box, which would supply, these are pneumatically actuated, supply air and control the timing and the actuation of these artificial muscles. So this is kind of a soft robotic diaphragm assist system, and it augments diaphragm function as a treatment for mechanical respiratory failure. This is an in vivo image, it's very quick, but as you can see, if we pressurize this artificial muscle, we can get downward motion of the diaphragm, and this is in a large animal model. This is the team that worked on this project. Lucy was my first graduate student from the group and really spearheaded this, and a really talented team of engineers and physicians. This is kind of what we looked at first. This is a cross-section of the lungs that are colored in red here, sagittal and coronal cross-section, and you can see the diaphragm is depicted here by the curves at the bottom of the lungs. You can see how it moves downward with each breath. So at the end of expiration, you have something like this, and then it moves at the end of inspiration, it flattens and contracts. And we thought we can develop something that will recapitulate that same motion, but augment it. So this is a very simple McKibben actuator. I'll talk later in the talk about how we've refined this, but we started with a simple McKibben actuator. At the end of expiration, the diaphragm is relaxed, and so the artificial muscle is unpressurized, and at the end of inspiration, it's contracted or pressurized. We did some large animal studies in a preclinical model. We used a swine model for these, and we worked at Boston Children's Hospital to do many of the initial studies and then moved them over here to MIT. A lot of instrumentation was required here, so we used a Power Lab system for data acquisition. We measured flow using the spirometry from a ventilator and volume. We also had a control box where we could trigger the actuation or the pressurization of our artificial muscles and time them with various mechanical and electrical signals that I'll describe a little bit more detail. And then we had multiple pressure sensors, so we could measure pressure in the pleural cavity and the abdominal cavity. And this was achieved with a transesophageal probe, where there was one pressure probe in the thorax and one in the abdomen. And all of this data was collected and synchronized together. We also used X-ray or fluoroscopy to image what was going on when we inflated and deflated these actuators. So you've seen this in vivo image of how the muscle creates this downward motion. There was a lot of work going into how we attach it to the rib cage and the sternum, and how many actuators to place and what kind of orientation to use them. Here you can see on X-ray, as the actuator is filling and emptying, you get this motion of the diaphragm. The air there is shown in bright, so you can visualize with fluoroscopy how it's moving. And then just from another angle, this is from a coronal cross section, you can see the heart here outlined in orange and the diaphragm underneath with each breath as it's moving. We created multiple models with various grades of respiratory insufficiency to test this in vivo. I'm just going to show very simply a vignette, like a piece of data from one of our studies. We've done over 12 of these. But here you can see when the system is on, and these are kind of, you know, the cyclical actuation we give to the actuators. When it's off is shaded in gray in the middle, and then when it's turned on again at the end. And if you look at the flow going into the lungs when the device is on, compared to when it's off in a model of respiratory insufficiency, and when it goes on again, you can see that we do achieve quite a difference in the flow in and out of the lungs. And here we have no mechanical ventilation. We're just using the native diaphragm function and the assistance from the actuators overlaying the diaphragm. Similarly, with volume, you can see how the tidal volume almost doubles when the assist device is on compared to when it's off. Another thing that we were really interested in is how this ventilator replicates and augments native respiratory pressures. So if you look at normal physiology, here's some idealized waveform for what happens during breathing. The pleural pressure goes down, the abdominal pressure increases slightly. Transdiaphragmatic pressure is the difference between them, and this draws flow into the lungs. When you depress this function slightly with either by, you know, anesthetics or by severing the phrenic nerve, you get the same waveforms, but they're blunted a little bit, so they're you know, there's not as much air moving in and out. With mechanical ventilation, which is the standard of care, you're pushing air in, and these waveforms look entirely different. So you're really changing the biomechanics of how the lungs are filling and emptying. This is a typical ventilator waveform. With our device, you really can get the same type of pressure and flow waveforms that you can see with native normal physiology. So that's pretty interesting. And we looked at things like the work of breathing. I won't go into that there, but, you know, we can maintain the same kind of biomechanics with this negative pressure ventilation. We found that timing is really critical here. So this is shown on the left here is when we're independently actuating our device and the diaphragm is moving separately, and then synchronized here on the right. And you can see how much more consistent the augmentation of flow and volume is. In this graph, these are different subjects or different porcine models, and you can see in each case synchronized actuation is a lot less variable and better overall in the increase in tidal volume that you see. So, you know, just to kind of summarize this one case study, we demonstrated an implantable ventilator that leaves the native diaphragm intact. We showed that we can augment diaphragm motion, respiratory pressures, and ventilation, and that we have this biomechanical advantage over mechanical ventilation. We're starting to move towards liberating the airway during mechanical ventilation for a specific patient population that have really progressive muscular degeneration. And this may allow patients to be mobile during assisted ventilation. Of course, there's a lot of ongoing work in how we control this best. So, you know, we looked at it, or we're now currently working on studies, ongoing studies, where we're looking at triggering using air pressure flow. We're looking at flow synchronization, and then we're also looking at EMG synchronization. So because we're already on the diaphragm, can we put sensors on the muscles of the diaphragm and trigger the actuation based on surface electromyography? And here we've performed a study where we've really looked at different parts of the diaphragm and where do we get the highest EMG amplitude so that we can trigger the device. And then the control system will be something like, you know, you sense the signal, you do some sort of filtering, and then you feed back into your electropneumatic regulator and microcontroller to get very synchronized actuation. We're also looking at improving the types of actuators. So I described a simple McKibben artificial muscle. Here we're looking at a laminated and wrapped and heat sealed type of actuator, bending actuator, which is more robust, more repeatable. We can model and we can get very specific bending, which correlates to diaphragm motion a lot better. This is also flatter and easier to insert, potentially minimally invasively. Oh, there's a question in the chat. Should I answer questions as I go? I can take this one. Speaking, singing, or swallowing food, do I anticipate that to be improved? Yeah, that's the goal ultimately, that we can do that. I think that takes a little bit more sophisticated control because there's big changes in, diaphragm motion during singing. But ultimately, we do want to move towards enabling those instead of, not just spontaneous breathing, but also yawning, coughing, singing, those kind of daily activities as well. Okay, we also had to develop really kind of high fidelity bench models to really look at how we can optimize this in the lab. So this is a simulator where we have a thorax and a dynamic abdomen where we can change the compliance of the abdomen. And then we can use that in the lab as shown here to measure changes in diaphragmatic function. We have a 3D printed rib cage we can attach the actuators onto. We can look at motion of the rib cage, really optimize it in vitro before moving to in vivo studies. Okay, I'm going to shift gears a little bit and talk about multimodal simulations of biomechanics. And here I'm going to give an example, two different examples. The first is the development of a high fidelity cardiovascular in vitro simulator. So here, what we're basically looking to do is to take heart tissue and reanimate it so that we have a beating heart on the bench and we can recapitulate all the biomechanics associated with it to lend itself to better testing of devices, better training, better education, better understanding. So we started off with this. This is the work of Clara, who was a graduate student in my group. We wanted to recapitulate biomechanics on a benchtop model. We saw a need because often heart models are either not recapitulating the biomechanics or not representing the anatomy, or doing one or the other. And we thought maybe we can use our soft robotic approaches to enable better benchtop simulators. And I have worked on a cardiovascular sleeve to augment heart function during my doctoral work. And then when I started the lab here, we thought maybe we can use the similar techniques, but to do it on the bench. So, great team again involved in this work. Really, this part was led by Dr. Park, who's now at Abbott. So we thought about taking an explanted porcine heart and really carefully dissecting the muscle out of it. And then replacing that muscle with a soft robotic cardiac muscle, joining them again then together with a functionalized adhesive so that we could actuate the synthetic part and reanimate it on the bench. This would give accurate anatomical details because we preserved all the valves, but tunable motion with electropneumatic control. So to do this, we dissected the muscle of the heart, we unraveled it, and there's this myocardial band theory, which helped us then to really get the orientation of the muscles. And we performed diffusion tensor MRI imaging to really visualize the muscle fibers. And then we built a soft robotic analog of that flattened ventricle, rewrapped it around the dissected heart tissue, and then we had, again, this 3D structure that we could animate. So this is kind of the heart in two different cross sections on a CT scan. The endocardial tissue is in lighter and the soft robotic muscles are darker. You can see how they're well coupled together, and this just shows these short axis and long axis view. We really wanted to use this model, so it's kind of cool Frankenstein type of heart that we had on the bench, but like how are we going to really use it and make it useful? So we did a lot of work on optimizing the design of the cardiac muscle, the synthetic part, and figuring out how to preserve the tissue so that then we could implement it in flow loops and use it for realistic device testing. Here's some details on the computational part. We used a thermal analogy and finite element model to apply this anisotropic strain to a soft robotic myocardium, idealized here. And we looked at ejection and we looked at isovolumetric contraction. We wanted to achieve physiological levels for both. We then could vary a lot of different design parameters, like the density of the actuators, the types of actuators, and the material properties of the matrix in which these actuators are placed. We looked at 3D McKibbin actuators, flat McKibbins, and flat pleated pneumatic artificial muscles, and then we could model the effect of each of these. Here we looked at a single layer designs, but we also looked at double layer designs where some actuators were circumferential and some were helical to achieve a twisting motion like the heart does. So with all these kind of permutations and combinations of layers and number of actuators and matrix properties, we optimized to have two different layers. One was flattened McKibbins and one was 3D helically oriented McKibbins, and they could achieve about an 80% volumetric displacement and also a rotation close to the physiological. And this is kind of the animation of the model with each cardiac cycle. You can see some rotation, and you can see that a lot of the volume in the ventricle is displaced. We also did some fluid structure interaction models to look at the fluid inside the chamber. And obviously, if this was blood, you wouldn't want to have stagnant blood. So we looked at the velocity with each contraction in an FSI model to look at areas of low flow. And you can see the velocity mapped here at different times in the cardiac cycle. So you really don't want anything down in the red zone when it's contracted, because you don't want stagnation, which can lead to clotting. So you can see with our optimized design, you get a lot less stagnant blood than with a control design, which just has 3 circumferential actuators. So that really helped us to optimize the design, and we made this or realized this in the lab. You can see a quite dramatic slow motion video here of the ventricle ejecting water here out of it when we idealized this. And you can see very little matrix material, which helped us, and then the circumferential and helical layers. Here you can see the pressure generated, so you get up to 120 millimeters of mercury, which is physiological, and about a 70% ejection fraction with each cardiac cycle, which again is a healthy kind of range. So then we attached it to a full flow loop, and we took this biorobotic hybrid heart, attached it to a flow loop where we could mimic arterial and atrial compliance and instrument it with multiple flow probes and pressure sensors. This is the entire setup in the lab with the control system data acquisition and the different compliance chambers. Now we could do interesting things like testing mimicking diseases and testing interventions. Here we can generate the aortic pressures, the left ventricle pressures, and the left atrial pressures very accurately. We can then visualize inside because we can perfuse with clear fluid, and we can look at the opening and closing of these tissue aortic valves, and we can image them with echo also. And we can look also at the mitral valve, and here's the mitral valve opening and closing. Then we can mimic disease. So if we cut some of the cords that attach to the leaflets of the mitral valve, you can actually get prolapse, and you can see some severe mitral regurgitation or leakiness or backflow. You can get both moderate and severe levels of mitral regurgitation. So here's baseline, moderate, and severe, and you can see how the regurgitant flow increases in each case. Then we said, look, can we mimic replacing the valve with the bioprosthesis or a replacement valve? So we put in this trifecta valve from St. Jude Medical, and you can see that pre-treatment, the leaflets don't really co-opt, you get leakiness, and post-treatment, you get this really nice coaptation of the leaflets. And if you quantify that with the sensors and the flow sensors in place, you can see a lot of regurgitation with severe MR, and with the valve replacement, you get back to baseline levels, similarly shown here with the regurgitant fraction. And we did this for a number of different types of mitral valve therapies, and these are ones that are really hard to mimic unless you go in vivo. So we ended up making a lot of progress in terms of research, and we're now looking at right heart models, models of congenital defects, mitral valve biomechanics, and we've started a lot of collaborations on this. We've also looked clinically at mimicking different types of valvular interventions and left atrial appendage closure with Boston Children's Hospital and Beth Israel Hospital. And we've also started some industry collaborations where we're using these models to test devices with Philips and Boston Scientific. So next step is to really looking at can we mimic things like myocardial infarction? What happens after that? What happens when you exercise? How does the heart remodel? If there's specific types of anatomical defect. We're looking at testing a variety of different interventions. And we're finally looking at, can we use these models to investigate flow and cardiac biomechanics? Okay, the next topic I'm going to cover is a biomimetic soft robotic sleeve that enables hemodynamic and biomechanical mimicry of left ventricular pressure overload and aortic stenosis. So the problem here was that there is no really good way to mimic aortic stenosis, a disease where the aortic valve becomes narrowed and doesn't allow enough blood through. We were looking to use soft robotics to mimic aortic stenosis with high controllability. This is a huge team that was helped with this work. We basically made a soft robotic sleeve with 3 different pockets that we could actuate independently to achieve different levels of narrowing in the aortic valve. This is what the sleeve looks like in an extensible layer on the outside and different pockets on the inside. We use this in a pig model again. We implanted it around the aorta, used MRI to measure the level of actuation, and then we used to measure cardiac function, and then we used catheterization to measure cardiac function with different levels of actuating of the sleeve. This is a typical, in blue, a typical pressure volume curve with each cardiac cycle. So the amount of pressure and the volume ejected with each cardiac cycle. And by filling one pocket, we can show we get higher pressure gradients across the valve, but less volume ejected. And that trend increases with selective or subsequent actuation or filling of each of these pockets. You can really get to a severe disease state with this. And this is done in vivo. So really nice way to tunably control pressure volume relationships in vivo. We can also use MRI to measure the flow through this sleeve depicting aortic stenosis and measure velocity with each different disease state. Next, we're looking at, and we've recently published work on using this sleeve in combination with a left ventricular sleeve, where we can change the stiffness of the ventricle and mimic the remodeling of the heart that happens with aortic stenosis in response to constant pressure overload. We can also mimic the profile in a patient-specific way, and these are in fully synthetic 3D printed models of the aortic opening, how it looks when we inflate this band to various degrees, and then we can mimic a changing stiffness or a decreased compliance of the ventricle with a patient-specific heart sleeve. Here we can mimic multiple different patient-specific CT effective orifice areas. We can also 3D print very patient-specific levels of calcification with multi-material 3D printing. And we can quantify how closely we're matching the effective orifice area with the DICE score and really closely mimic. Here we show 13 different patients, some with stenosis and some with actually congenital heart disease. And when we use the multi-material 3D printing, we can do the same. These are what the ventricular sleeves look like, and you can see with using this sleeve, we can create pumping motion in a synthetic benchtop model. So we can recreate both the contractility, but also the stiffness changing of these ventricles in the lab. Here I show the compliance reduction over time as you can increase the stiffness, and we can show these changing end diastolic pressure volume relationships when you have a healthy heart, and then to moderate and severe remodeling. We can also mimic the effect again of placing a valve replacement. So before and after, you increase the effective orifice area and you decrease the high pressure gradient that is formed around this, across this this stenosed or narrowed aortic valve. There's a nice video here that I will show to finish, and then I'll
The size and shape of the human heart can vary from one person to the next. And for people living with heart disease, these differences can be particularly pronounced. A team of MIT engineers are hoping to help doctors tailor treatments to a patient's specific heart form and function with a new, soft, custom robotic heart. The team has developed a robotic system to control soft 3D printed replicas of a patient's heart that can be actuated to mimic the patient's blood pumping ability. The procedure involves first converting medical images of a patient's heart into a three-dimensional computer model, which the researchers can then 3D print using a soft polymer-based ink. The result is a soft, flexible shell in the exact shape of the patient's own heart. To mimic the heart's pumping action and the motion of the aortic valve in disease, the team has fabricated sleeves similar to blood pressure cuffs that wrap around a printed heart and aorta. They tailored each sleeve's pockets such that when wrapped around their respective forms and connected to a small air pumping system, the sleeves could be tuned separately to realistically contract and constrict the printed models. By using patient-specific data, the researchers are able to accurately recreate the same heart pumping pressures and flows that were previously measured in a specific patient. Doctors commonly treat heart diseases such as aortic stenosis by surgically implanting a synthetic valve designed to widen the aorta's natural valve in the patient's heart. This new system, developed at MIT, would allow doctors to first print a patient's heart and aorta, then implant a variety of valves into the printed model to see which design results in the best function and fit for that particular patient, informing specific surgical decisions and procedures. In the future, the researchers say these heart replicas could also be used by research labs and medical device industry as realistic platforms for testing therapies for various types of heart disease.
All right. Yeah, so hopefully from that video, which was much more eloquent than I can describe it, you can see how we can recreate a realistic aortic flow patterns to model both aortic stenosis and congenital valvular defects. Really look at high fidelity models of cardiovascular disease for hemodynamic mimicry with soft robotics and patient-specific modeling. This can help to identify patients that are suited for different clinical interventions and really help to test and develop novel therapies. So with that, I'll just remind you of this kind of process, and hopefully it's clear that by spending time in this testing loop here, we can really give a lot of good insight back to the design and the clinic and to industrial collaborators. And I thank you for your time. This group has been a fantastic group to work with in the last close to 6 years, and thanks for listening.
Thank you so much, Professor Roche. That was an amazing and inspiring talk, as always. I don't know, do you have time for any questions now, or are you, do you have to run up?
I probably need to run, but I'll rejoin the discussion session later if that.
Oh, excellent.
Or I'm
okay.
happy to answer any questions over email as well.
Okay, yeah, or Discord, I'm sure. Okay, well, in that case, I will just applaud for everyone. Thank you so much, and good luck teaching.
Thank you. Bye.